Researchers, particularly a team at Stanford University, have successfully genetically engineered yeast to produce opioid compounds from sugar. This was achieved by introducing a complex set of genes up to 23 from plants, bacteria, and even mammals into baker's yeast. These genes enable the yeast to convert simple sugars into opioid molecules like hydrocodone and thebaine, which are typically derived from the opium poppy. This scientific breakthrough means that, instead of relying on the lengthy and resource-intensive process of cultivating and processing poppy plants, it is now possible to produce these pain-killing medicines in just a few days using yeast fermentation. The process involves the yeast acting as a miniature factory. When fed sugar, the engineered yeast carry out a series of biochemical reactions, ultimately yielding opioid compounds. Currently, the amount of opioid produced by these yeast strains is very small and not yet commercially viable, but the proof of concept is significant. The technology could eventually lead to faster, more controlled, and possibly less expensive production of opioid medications. However, it also raises concerns about the potential for illicit drug production using similar methods, since the process could theoretically be adapted outside of regulated environments. In summary, genetically modified yeast can indeed convert sugar into opioids, and this represents a major advance in synthetic biology and pharmaceutical manufacturing. To produce lysergic acid derivatives like LSD in yeast, researchers have managed to reconstruct the entire biosynthetic pathway for D-lysergic acid, which is the key precursor to LSD inside baker's yeast. This is done by introducing a set of genes from ergot alkaloid-producing fungi into the yeast genome. These genes encode enzymes that direct the yeast's metabolism to synthesize D-lysergic acid from simple sugars, essentially copying the natural biosynthetic process found in fungi. The main genetic modifications involve adding genes such as DMAW, ESF, ESC, ESE, ESA, ESD, and ESG. These genes work together to convert tryptophan and other basic building blocks into the ergoline scaffold and ultimately into D-lysergic acid. To improve production, scientists also adjust promoter strengths, increase gene copy numbers, and modify certain yeast genes like FAD and PDI1 to enhance protein folding and cofactor availability, which helps the introduced enzymes function better. More advanced strategies include targeting some enzymes to specific parts of the yeast cell, such as moving ESA-C to peroxisomes,